Hello everyone and warm welcome to all of you. This is Chapter 11 Part 2. This part will include osmosis, significance of osmosis, plasmolysis, 𝕆𝕆𝕋. Then we have to see absorption of water by roots, that is through apoplast pathway and 𝕆𝕆 pathway. This is Chapter 11 Part 2. This is Part 2, Chapter 11 Transport in Plants. First we have to discuss here osmosis. In your earlier classes also you are acquainted with term osmosis. That is, plant cell is surrounded by cell membrane, and cell wall. If this is a plant cell we have, then this is the cell wall. Cell wall is permeable to all, and below cell wall we have that is plasma membrane. Cell wall is freely permeable while this plasma membrane is selective permeable. In plant cell we have that is vacuole. We have the membrane of the vacuole is known as called 𝕆𝕆𝕆. You must remember that is central vacuole surrounded by this 𝕆𝕆𝕆. So in plant cell, the cell membrane and the membrane of the vacuole, that is 𝕆𝕆𝕆, these are important determinants of movement of molecules in and out of cell. What is osmosis? We have to see the term is used especially diffusion of water across a differential or selective permeable membrane. Plasma membrane is selective or differential permeable. Osmosis occurs spontaneously in response to a driving force, and osmosis that depends on pressure gradient, and that is concentration gradient, right? That is concentration gradient and pressure gradient. We have to say that if water moves from region of higher concentration, higher water potential, to the region of lower concentration until equilibrium is obtained. You see, in osmosis, if here that is the two solutions are separated by this is semi-permeable membrane. Let here we have these are the solute molecules, and here we have number of the solute molecules is more. So we'll say that this solution is dilute one. This is concentrated solution. In term of solution, this is dilute and this one is concentrated one. But in reference of water molecules will be more here. So this is a special type of diffusion where only water molecules, they will move from a place of their higher concentration to low concentration, or we can say they will move from dilute concentration or dilute solution to concentrated solution. How can we demonstrate osmosis? One is potato osmometer. Simply what we have to do in this osmometer, we have to do this one. We have to take a large size potato. We will peel out this potato. Outer skin will be removed. The bottom will be flattened, and here we will make a cavity inside. And now that will be kept in a trough here. Here in this we may fill concentrated solution of sugar here. Here the solution is dilute. This solution is hypotonic. Here the solution is hypertonic, concentrated one. What will happen? The cells in between, millions of the cells will be there, and these cells will permit osmosis. That is, in this cell the water will move from higher concentration of water to lower concentration. Water molecules will enter here, and in that way water from all the sides from this trough ultimately will accumulate here in this cavity consisting of hypertonic solution, and in the last the level of this hypertonic solution will rise. We can demonstrate this by Thistle funnel experiment also. In Thistle funnel, that we will discuss in diagram. What is there in Thistle funnel? This is the Thistle funnel, inverted funnel. This is with the long neck. Here we have concentrated solution of sucrose, and here we have this is semi-permeable membrane. It is kept here in water. Water is hypotonic one. This one is hypertonic one. Osmosis will take place. Water molecules will move from outer to inner side, and because of this the pressure will be generated, and because of this the initial level which was here in both outer and inner solution, now it will increase. That increase is because of osmotic pressure. Osmotic pressure, how can we define? It is a pressure which is exerted by inward movement of water molecules from hypertonic solution to that is concentrated solution. You see here, first this is funnel, a solution of sucrose in water, is it? That is a funnel separated from pure water in a beaker by selective permeable membrane. How to get this selective permeable membrane? That is membrane of an egg is used, yolk is removed, albumin, that is a small hole that will be taken out, and it will be placed in dilute solution of hydrochloric acid for few hours. What will happen? Eggshell being made up of calcium carbonate will dissolve and the membrane will remain intact. This membrane we can that is put on the mouth of funnel and it will become that is semi-permeable membrane. Now osmotic pressure, I told you because of osmosis the level will go up, and that is because of osmotic pressure exerted in this solution by movement of water molecule. If we put a pressure on this so that the level of the water molecule doesn't rise in hypertonic solution also, then this pressure that will be known as osmotic pressure. This osmotic pressure, how can you define? That is the pressure exerted from outside which will prevent the inward flow of water molecule when the two, that is hypertonic solution, is separated from that is either dilute solution or by its pure solvent. Is it clear? So numerically osmotic pressure is equivalent to osmotic potential, but here the sign is opposite. Osmotic pressure is having plus sign and the osmotic potential it is having that is negative sign. Okay, come to the next now. Now very important and interesting part we have here. You see the two solutions are separated here. You see solution A and solution B. You see here these are the solute molecules. These are the solute molecules. More solute molecule we have this side. It means in layman term we can say that this solution is that is dilute solution, while we can say that this one is that is concentrated solution. This one is it right? This is a selective permeable membrane. This one is. What do you think? That is, this is a dilute solution is separated from concentrated one, or if we may assume that that may be pure solvent which is having high water potential. Here the water potential of this will not be zero definitely, because zero water potential will be a pure solvent. That this is 1, 2, 3, 4, 5. Let its water potential is, let this water potential is minus 5. What is the water potential of this? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13. Let its water potential will be minus 14. So which is having more water potential? A is having more water potential. The water potential of B is less. The water will move from that is high water potential to low water potential. Is it very much clear to you? Yes. Now some questions we have here. The two chambers A and B containing solutions are separated by semi-permeable membrane. Solution of which chamber has lower water potential? Chamber B. Solution of which chamber has lower solute potential? This decrease in water potential is because of addition of solute. So here that is solute, more solute is added. That's why the solute potential of this will be almost equal to minus 14, and here that will be equal to minus 5. So here the answer will be that is B. In which direction osmosis will occur? From high water potential to low water potential, from chamber A to B. Which solution has high solute potential? Obviously this is having high solute potential. At equilibrium what will happen? Which chamber will have lower water potential? No, when equilibrium will be there, both chamber will have that is same water potential. You see one chamber has that is minus 2000 kilopascal, that is other is having minus 1000 kilopascal, that is water potential. So what we can say, that is which is higher one? That is chamber with this is higher water potential. Is it clear to you? So this is NCERT diagram. Frequently based on this the questions are coming in examination. Come to the next now. Proceed to plasmolysis. Plasmolysis, that is also related to osmosis, occurs when the water move out of the cell and cell membrane of the plant cell shrink away from the cell wall. That is called plasmolysis. That is when a cell or tissue is placed in hypertonic solution, has more solute, that is to the protoplasm, water will move out. It is first lost from the cytoplasm, then it is lost from vacuole. Important question. Water loss from the cell, first it will be from cytoplasm, then it will be from vacuole, and the cell will be said as plasmolized one. If this is the cell, this is plant cell 1. So here if it is placed in that is hypertonic solution, then the water will be lost from the cell, and ultimately that is this cytoplasm or protoplasm will be concentrated at the center. That will be movement of water that is occurred across the membrane, that is an area of higher water potential to an area of lower water potential. You know it, the same thing. Space between here, that the space here, that what will be occupied by external hypertonic solution, because the cell wall which is here, this cell wall is permeable to this hypertonic solution. In the last, this hypertonic solution will enter, and that will be in between the cell wall and that is plasma membrane. When the cell is placed in isotonic solution, there is no net flow of water towards the inside or outside, and that is solution is known as, that is, it is isotonic to that is cytoplasm. When water flows into the cell and out of the cell in equilibrium, that stage is known as flaccid one. Is it? Like this is a plant cell here. So here that is amount of the water which goes in and which goes out, if is equal, the cell is known as flaccid. Stage of plasmolysis, you see children, the three stages are being identified. One is limiting one, followed by incipient plasmolysis, and followed by that is evident plasmolysis. This is clearly visible plasmolysis is evident one. What is there in limiting plasmolysis? You see, characterized by reduction size of the vacuole, water will be lost from cytoplasm and vacuole due to osmosis, water goes out, first stage, and that is here gradually the water is being lost. The turgor pressure is 0. Remember, turgor pressure is 0. Protoplast is in contact with the cell wall but doesn't press the cell wall now, therefore the turgor pressure is 0. It continues. If it is kept in hypertonic solution, this process continue, then the cell wall cannot contract more, therefore the contracting protoplast withdraw it from cell wall, and here that is cytoplasm start to leave the cell wall here, especially at the corners, that is corner the cell wall and the plasma membrane, they will get that is detached. Here we can identify them separately, and here the turgor pressure of the cell will decrease, that becomes negative. If the process continue even now, that is known as evident plasmolysis. Final stage of plasmolysis, continuous contraction of the protoplast, and it becomes nearly rounded to the, that is at the center, cannot remain alive for a long time, and here the turgor pressure will become more negative. Remember, I told you that the turgor pressure or that is pressure potential, this was positive, but in this case in a plasmolyzed cell, that is this turgor pressure or pressure potential will be negative one. Important question this is. Now deplasmolysis. Plasmolysis, you know, this is a reversible process. That's why if a plasmolyzed cell is kept in hypotonic solution, then the water will enter due to endosmosis, that is again the cytoplasm that will regain the normal shape. The turgor pressure will increase. The pressure exerted by protoplasm due to entry of water on the rigid cell wall will be known as now that is pressure potential. Is it? Because of rigidity of the cell wall doesn't rupture the turgor pressure, ultimately responsible for enlargement and extension, that is growth of cell. Important. Plasmolysis is property only of living cell. An interesting question came once that a teacher wanted to demonstrate plasmolysis, and she took that is a preserved specimen, that is specimen of 𝕆𝕋𝔼𝕆. You know, preserved specimen of 𝕆𝕋𝔼𝕆, that will not be living cell, that is a dead one. In dead cell, the cell wall as well as that is plasma membrane and tonoplast, all they will die, they will become permeable. That's why we cannot demonstrate plasmolysis in that is dead cell. Application of plasmolysis, that is if you want to identify the cell is living, that will be plasmolyzed. method for measurement of osmotic pressure. If there is only incipient plasmolysis in a solution, then the concentration of this solution will be almost equal or little higher to that of cytoplasm. Incipient plasmolysis, that is helpful to determine osmotic pressure. Food preservation, that is jams and that is jellies and that is pickles. These are being preserved because of plasmolysis. If any that is unwanted bacteria or fungus spore will come, instantly it will be plasmolyzed and it will not grow. Elimination of weed. In the root of weed, if we will put some that is concentrated solution of that is sodium chloride, that is the exosmosis will take place and the weed may die. Right? Now we have to see the next one, that is imbibition. Imbibition, it is also a special type of diffusion, that is in which the water is absorbed by solid colloids, water loving substances these, and because of this they will increase in that is volume. Classical example of water absorption, that is by seed. When the seed is kept in soil, it will absorb the water, and the seed inside is having water loving substances. The seed coat will burst and that is the seedling will come out. It is because of imbibition. The pressure produced by swelling of foot in prehistoric time, if that is this is the rock, a fine crack is there. In this crack the dry wood will be kept here. In rainy season it will absorb, imbibe the water and it will generate a pressure, that is it. Seedling have been able to emerge, I told you just now. Imbibition is also due to diffusion, that is concentration gradient is there, is it? Water potential gradient is established between absorbent and the liquid to be imbibed. This is according to higher concentration, higher water potential to lower water potential. And in addition, any substance to imbibe any liquid, affinity is required. For example, that is rubber is having affinity with that is alcohol, and similarly the wood is having that is affinity with water. If wood will be kept in alcohol, they do not have affinity, that's why imbibition will not take place. And the last, power of imbibition is variable. Maximum imbibition is found in agar and agar, then pectin, then protein, then starch, then in cellulose. That is. Come to the next now. We have to see now long distance transport. By now we have discussed small distance transport. In case of that is plant, this is the plant here, this is the root, substance that is water and minerals are absorbed by that is root and they will be transported to that is tip of the stem very very far, long distances. Similarly, the leaves will make that is photosynthetic products. These photosynthetic products are required by tip of the plant, by that is other leaves and that is other part of the plant, and sometimes that is root when the roots are that is functioning as storage organ. Otherwise food is required by all part of the plant, either this is this side or this is this side. But this long distance movement, it cannot be because of that is diffusion only. Diffusion is a slow process, but you have discussed. It occurs only for short distance. One can explain, for example, movement of water molecule across a typical plant cell. If this is typical plant cell and water want to move from this side to this side, that is typical plant cell, it is about 50 micrometer. It will take about 2.5 seconds. And you see that if from root to tip of the plant, trillions of the cells may be there, even more than that, it will take a lot of time. And therefore we have special long distance transport system which is necessary to move substances across long distances much more faster. Water and minerals and food, I told you here, generally move in mass or bulk flow. Mass flow is the movement of substances, that is NMC, from one point to the another point, as a result of pressure difference between the 2 points. We have to discuss this thing in detail, and that is here what will happen, just like that is suspension are swept along the same pace as in flowing river. That is, a flowing river is there, substance is put at one end and it will be transported to other end, which is very, very away from the point where the substance is being input. Now it is achieved by positive hydrostatic pressure, that is from a place from where it is to be transported. It is having positive hydrostatic pressure, higher pressure, just like a garden, that is house we have, or a negative hydrostatic pressure will be at, that is the pressure where the substance is to be transported. The bulk movement of substance through conducting or vascular tissue is called as translocation. And you know, two type of vascular tissue we have, one is xylem, one is phloem. Xylem is associated with the translocation of mainly water and minerals, salts, some organic nitrogen, hormones from root to the aerial part of the plant. On the other side, you all know phloem will translocate a variety of organic and inorganic solutes, mainly from leaves to other part of the plant. It may be even, that is, storage organs. Is it? Now you have to see one by one, that is absorption of water by plant. Absorption of water by plant we have to see now, that is root absorbs most of the water. Is it? Responsibility of the absorption water and minerals is more, especially function of the root here. They absorb the water and minerals which are present in millions at the tip of the root. These are thin-walled, slender structure extension of the outer layer. Water is absorbed along with minerals solute by root hairs purely by diffusion. Once the water is absorbed by root hair, that they will move to the deeper layers in two distinct pathways. One is, that is apoplast, one is 𝕆𝕆𝕋. Apoplast, see, that is the system of adjacent cell wall that is continuous throughout the plant except Casparian strips of endodermis in the root. Apoplastic movement exclusively through, that is intercellular space and the wall of cells. It doesn't require to crossing of the membrane. Movement is dependent on gradient from high water potential to low water potential that will move. Apoplast doesn't provide any barrier. The things will move faster rate. See this diagram? This is symplastic, this is apoplast. You can see this is apoplastic one, cell wall and the intercellular space. Water molecule will enter here and they are moving to the, that is apoplastic movement. This is endodermis, this is Casparian strip, and that's why up to Casparian strip water molecules will move by this apoplastic movement. Symplastic pathway, that is system of interconnected protoplast. Neighboring cells are connected through cytoplasmic connections known as plasmodesmata, right? And that's why, that is the water or the substances, they have to move through cytoplasm. Then intercellular movement is through plasmodesmata, has to enter the cell through cell membrane, hence the movement is very slow comparatively. Movement is again down a potential gradient. Symplastic movement may be added by cytoplasmic movement. Most of the water in a root, it is through apoplast. It moves through apoplast of cortical cell because these are loosely packed and they do not have more resistance. Inner boundary of the cortex, that is endodermis, it is impervious to water and it has Casparian thickening which doesn't allow, that is the movement, symplastic movement. Water molecules are unable to penetrate this layer and they are directed to wall regions which are not submerized, that is through passage cell the water will enter into the xylem. The same diagram we have, this is the root hair we have here, that is, this is the symplastic movement. It is going through cytoplasm, it is going through membrane and it has entered into the endodermis cell. Now this is passage cell, through passage cell it has entered into the xylem. This is apoplastic one, that the water molecules that is moving through cell wall and intercellular space and ultimately it has entered here. Symplastic and apoplastic pathway this is about, that is how the water will move, will absorb by root, apoplastic and symplastic movement. Okay, thank you.